So in the last video, we talked a lot about how red blood cells or erythrocytes are born. Now it's time to talk about their end of life because they don't live that long, right? We said about 120 days. And they're basically sacs of hemoglobin. So at the end of the life of a red blood cell, what you want to do is recycle the hemoglobin. And hemoglobin, as we mentioned, is a set of polypeptides, proteins, right? So if you break those apart using enzymes, you create amino acids, and those are useful. So we can just send those back into the blood supply. The tricky part is that in addition to those polypeptides, you have in each hemoglobin molecule 4 heme groups with a little iron molecule in the middle. Now the iron molecule can also be reused. So we bind it to a plasma protein called transferrin, and it gets taken generally back to the red bone marrow where it can be used to build new hemoglobin. But the rest of that heme group can be toxic as it breaks down. So you don't want old red blood cells just becoming stiff in the membrane and rupturing just anywhere in your body. So when they start to get old, generally in the spleen especially, occasionally the liver, the bone marrow, you have macrophages which will engulf these old red blood cells and begin this process of recycling them. So what happens to these heme groups? Well, heme is turned into something called biliverdin, which is turned into bilirubin, which is then released into your bloodstream. And it's slightly less toxic than the biliverdin or the heme. And that, though, needs to be taken out of your bloodstream by your liver, and then it gets packaged up with some other stuff, and it becomes bile, and it gets sent from your liver into your intestines. So your liver takes bilirubin out of your blood, turns it into bile, puts it in your intestines. Now, bilirubin is a yellowish pigment. So if you have too much bilirubin in your body, you kind of get a yellowish tint to your skin, in your eyes. You can see yellowish tint, and that can be an indicator that your liver is not functioning well. So if you notice that you are consistently turning a little bit yellow, it might be time to talk to your doctor. Now, this is frequently something that occurs in babies, and it's not necessarily a sign of liver failure in babies. It's pretty common because babies have been making red blood cells, right, since they were conceived, or shortly thereafter at least. And so that's longer than 120 days. We've had dead red blood cells we've had to recycle. And so we've had heme groups that we've had to turn into bilirubin. And all of that gets sent back into your intestines as bile, but they don't poop yet, right? They can't be pooping very much until the very end when they want to starve their environment and fill it with poop. So their intestines fill up with bilirubin. A lot of that gets reabsorbed into the bloodstream, and so babies can have high bilirubin levels sometimes when they are born. Now, usually the answer to this is get them pooping, right? So that usually means get them eating. So nursing and pooping will fix that. UV radiation can also break down bilirubin, and so sometimes they tell you to get them by the window or out in the sunshine, or they'll put them under a lamp or in a blanket that puts out UV radiation, and usually it's not that big of a deal. So as a general summary, you produce new red blood cells in the red bone marrow after 120 days, give or take. Those are broken down by macrophages in the spleen. We recycle those old hemoglobin molecules into amino acids, iron, which gets sent back to the red bone marrow, and heme, which gets turned into biliverdin and then bilirubin, which is then taken out of your body at the liver and put into your intestines as part of bile. Now, it's worth following what happens to your bilirubin once it gets into your intestines, because your intestines is filled with bacteria. And these bacteria are looking for food, things with nutritional content like bilirubin. And so they will metabolize the bilirubin, and it will become converted into urobilins and stercobilins, which are pigments that are yellow and brown. And that is why your poop is brown. Congratulations. You now have a new, maybe not dinner conversation point, but interesting thing to talk about with friends. And If you have liver problems, you can have white poop, right? If you're not putting bilirubin into your intestines, you can end up with white poop. It's wild. Now, because your intestines is reabsorbing stuff, right, nutrients, right, some of those urobilins get reabsorbed from your intestine back into your blood, and they get taken out of your blood by your kidneys, which filter your blood, right? And those urobilins, which get filtered out of your blood into your urine, this is yellow pigment that gets filtered into your urine. It's why your urine is yellow. Today you learned that red blood cells, when they get old, are the reason that your poop is brown and the reason that your urine is yellow. Same reason. Congratulations, you now have a story to tell. And this is a summary of that basic process, right? Bilirubin gets taken out of the blood, put into your intestines, turned into urobilins and stercobilins, which make feces brown. Urobilins get absorbed into the bloodstream, which get taken out by the kidneys and get eliminated in urine, which is then yellow. And who told you that A&P wasn't interesting? Now, it's very likely that you are familiar with the idea of blood types. Blood types are determined by an antigen or a series of antigens that are found on the surface of your blood cells. Now, antigens are just, glycoproteins, combinations of sugar and protein, or sometimes glycolipids, that are genetically determined and they are sort of a signal to your immune system, like an ID card to your immune system that says, I'm part of your body. I am not an invader. Please don't bother me. If something with an antigen that your body is not familiar with enters your body, your immune system will often recognize that, and it will cause a big immune response that will fight it off. And we'll talk about that more when we get to the immune system. This stuff that we're talking about now will be relevant at that point. But this is a little bit different than your typical antigen-antibody complex. And while there are lots and lots of blood antigens, there are three that we talk most about: A, B, and something called the Rh factor, or sometimes it's called D. Now, Rh factor you may or may not have heard of, but you will have heard of positive and negative, right? If someone is positive, it means they have the Rh antigen. If someone is negative, it means they do not have the Rh antigen. Now, ABO blood groups and the Rh antigen are similar, but they're on two different genes and they operate a little differently. So let's start with your ABO blood groups. A is a particular type of antigen, and if you have the A antigen on your blood cells, then you are type A blood. If you have the type B antigen, then you are type B blood. If you have genes that give you both the type A and the type B antigen, both on your blood cells, then you are type AB. And if you have neither of those antigens, then you are type O. Now, whichever antigens you have, right, you are going to not have those antibodies, because antibodies are roving around looking for antigens that are foreign to your body so that they can attack them, essentially. So if you are type A and you have the A antigen on your blood cells, you do not want to have the anti-A antibodies. That said, you will have the anti-B antibodies. If you're type B and you have the B antigen on your red blood cells, you do not want to have the B antibodies, but you will have the anti-A antibodies. So you essentially have the opposite antibodies of whatever antigens you have. What if you have both antigens, A and B? Well, then you're going to have neither antibodies. You will have no antibodies for this set of antigens. If you're type O and you have no antigens, you will have both antibodies, both A and B. And each of the antigens have a particular shape, right? And the antibody, anti-B antibodies, will bind to the B antigens, again, to attack them. The anti-A antibodies will bind with their shape to the A antigens and attack them. So again, while you do not want to have matching antibodies and antigens in the same body. And so you won't, right? If you are type A, you will never have anti-A antibodies. And it is also why we have to be careful with transfusions. You can never give blood. So when you give blood, you're giving the red blood cells generally. You can never give your red blood cells to someone who has antibodies that will attack those red blood cells, because that will cause them to clump and then be destroyed, and then you release those heme groups into your body, and as we talked about, that can be bad for you. So if you have type A blood, you can never give to someone who's type B, because they have these B antibodies. You can give to someone who's also type A because they don't have those anti-A antibodies, right? You can also give to someone who's AB because they still don't have those anti-A antibodies, right? You cannot give to O or B because they have these anti-A antibodies. So essentially, if I'm type A, I can give to anyone who also has the A antigen because I know they won't have those A antibodies. They can have other antigens too. They can be AB. It's fine. AB positive, AB negative, whatever. It doesn't matter as long as they have that A. But if someone doesn't have that A antigen, then they will have that A antibody. So people who are B, people who are O, then that's going to be bad news if I donate to them. The donor's antigens and the recipient's antibodies cannot match up. So what if I'm the recipient? Let's say I'm type A and I'm the recipient, right? Well, what matters is their antigens and my antibodies. What antibodies do I have if I'm type A? Anti-B. Anti-B will attack the B antigen. So I can get blood from anybody unless they have the B antigen. So can I get from other people who are A? Yes, there's no B antigen. Can I get from people who are B? No, my anti-B antibodies will attack their B antigen. What if someone who's AB? No, because they still have that B antigen and I will attack it. Someone who's O? Well, yes, I can get from somebody who's O because they don't have a B antigen for me to attack. In fact, O, specifically O negative, is the universal donor of red blood cells. O negative can give to anybody. So what precisely happens when you get mixing of the same lettered antigens and antibodies, right? We know it's bad. Well, what happens? Well, the antigens, which are also called agglutinogens, right? When they're exposed to the corresponding antibodies, which are also called agglutinins. So let's say the A agglutinogen or antigen meets with the anti-A antibody or agglutinin. Those antibodies will grab onto those antigens, but they won't just grab, they have multiple binding sites, right? So you can grab onto the antigen on one blood cell and another antigen on another blood cell, and now these blood cells are connected, right? And the same thing could have happened with this blood cell and another because of a different antibody. Anyway, all these blood cells clump up, and it's called agglutination, right? And this not only clumps them up, which obviously can cause clotting and backups in your bloodstream, but also it marks them for destruction, and so it can cause this hemolysis, which again explodes all of this hemoglobin into your blood, which is not good when it's not bound within a cell because those heme groups can be toxic. And this is essentially what that looks like, right? These antigens are bound to by the antibodies and it clumps them all up, and then they may or may not get hemolyzed. Now, Rh is similar, except it's on a different gene, and there are a couple of small differences. So Rh is Rh because it was discovered in rhesus monkeys, and if you have the Rh antigen, we call you Rh positive. If you don't, you can be Rh negative. So your whole inclusive blood type with the ABO blood group and the Rh group is going to be like O negative or A positive or AB negative, something like that. Now, one way you can tell what type of blood type someone is, right, is by drawing their blood and mixing it with the antibodies, with known antibodies. So let's say I draw some of your blood, and I have no idea what your blood type is. I don't know what antigens you have on your red blood cells, right? Well, I can mix your blood with anti-A antibodies and see if it agglutinates or clumps. If it does, that means that there must have been A antigens in your blood to clump with the anti-A antibodies that I put into your blood, right? That means you must have type A blood or type AB, right? Now, let's say I put anti-B antibodies into your blood and it doesn't clump. Well, that means you must not have had B antigens in there to match with the anti-B antibodies. And so that means you must have had A, but you didn't have B, so you're type A. And then lastly, I will take another sample of your blood and I'll mix it with the anti-Rh antibody and see if it clumps. Let's say it does, right? So it clumped when I mixed the A antibody. It didn't clump when I mixed the B antibody. It did clump when I mixed the Rh antibody. That means you had A and Rh antigens. You are A positive. Now, this is different from talking about transfusion reactions, who can donate to whom. This is just figuring out your blood typing. But knowing your blood type is important to knowing to whom you can give a transfusion.